In this section, we will look at triacylglycerols. Glycerols are simply the esters of glycerol with fatty acids. So to illustrate, we start with glycerol with its 3 alcohol groups and 3 fatty acids. Again, here just labeled R1, R2, and R3 for their hydrocarbon tails. So we just form an esters among them. So it's a triacyl glycerol. Tri, 3, acyl, the residue left from your carboxylic acid, and glycerol, which is our parent alcohol. Triacylglycerols are also known as glycerides or sometimes triglycerides. In terms of their structures, they can be neutral glycerides. It means that they don't carry any charges. So it's nonpolar. There's no overall charge on the molecule. They can be monoglycerides, diglycerides, or triglycerides, depending on, guess what? The number of fatty acids that are esterified into your glycerol. Now, for phosphoglycerides, also known as phospholipids, it's made up of a diglyceride plus a phosphoryl group. Okay, so you now have a phosphoglyceride. This one that is now amphiphilic because the phosphoric acid group that is connected to the third alcohol of your glycerol usually carries a charge. Okay, so it's a polar phosphoryl group and a nonpolar fatty acid component. So it's amphiphilic, meaning it can be attracted to both water and nonpolar solvents. So let's look at neutral glycerides. Okay, so a monoglyceride, as mentioned, it only has one fatty acid connected to the glycerol. Diglycerides made up of 2 fatty acids. And this is a very important component of membrane lipids, more specifically the phosphoglycerides, as we will see in a later presentation. So structure of a diglyceride, but it doesn't usually exist as simple diglycerides. Okay. Triglycerides is the one that we are more probably the one that we think of when we think of lipids. Now, from our everyday experience, it's a glycerol plus 3 fatty acids, and its main function is as an energy storage. This is the fat deposits in our fat cells. So structure of triglyceride. Okay. So how are glycerides named? The systematic way is to indicate the fatty acid name, but using the oil ending, indicating that this is now just the acyl group of your uh, carboxylic acid, if you recall your organic chemistry, and then just add glycerol, which is our parent alcohol. So example, 1 palmitoleoyl, 2 linoleoyl, 3 stearoyl glycerol. It means that again, you start with your glycerol, and in carbon 1, you add the palmitoleil um, component, just the acyl component of palmitoleic acid. Linoleoyl means that again, the linoleic acyl component is connected to carbon 2. And stearoyl, you have now the acyl component of stearic acid connected to carbon 3 of your glycerol. If you don't have a mixed glyceride, meaning all 3 amino acids, all 3 fatty acids are the same, then you can simplify it as tristearoyl glycerol. And then sometimes they have common names. For example, the tristearoyl glycerol is also known as tristearine. Okay, so this is a structure of tristearine. It's a simple triglyceride. Mixed glycerides, this is the structure of what was mentioned earlier. It's a mouthful, so I will not say it anymore. If you recall from your organic chemistry, esters undergo hydrolysis. But glycerides are simple esters, okay, except that again, your alcohol is glycerol and your carboxylic acid are fatty acids. So it can undergo base catalyzed hydrolysis, also known as saponification. You may have performed this experiment at home or in the lab for maybe some of you uh, from your organic chemistry laboratory. Okay, it's simply really just the ester plus water plus base gives you the carboxylate plus the alcohol. Okay, and our alcohol, again, I keep saying is glycerol. So here's the generic reaction, recall from your organic chemistry. And then the carboxylate group that precipitates is the one that we use as soap, hence saponification. Now, sapon refers to soap. However, recall that one problem with carboxylate groups is that they precipitate with calcium and magnesium ions, and they form the scum that you may observe in your bathroom tiles after taking a bath. Uh, it can also undergo acid catalyzed hydrolysis, but the base catalyzed hydrolysis I think is more common. But if you have acid-based catalysis, acid-based catalysis, then you get the carboxylic acid instead of the carboxylate. Okay. Triacylglycerols. Okay, so if we now use the triglyceride as a source of energy instead, then we have enzymes, lipases or lipases that are being used. So we'll start with the one on the right. The one described in the previous slide was the saponification reaction of the triglyceride to form the sodium salt of the fatty acid, and this is the one that we use as a soap. Or again, in organisms, the fatty acids are metabolized by enzymes. So it undergoes enzymatic hydrolysis using lipases, releasing glycerol and the ionized fatty acid, and then used as the case may be. So it's usually in that case is usually for energy production. Okay, as previously mentioned, 1 gram of triglycerides or fat is equivalent to 9 kilocalories or 9 capital letter C calorie, or again, if you want to be SI about it, 37 kilojoules. So let's just distinguish between fats and oils. There's really no difference between them chemically. It's except that fats are the triglycerides that are solids at room temperature. Okay, so they are usually made up of saturated fatty acids. Recall our discussion on the effect of unsaturations or saturations on the melting point earlier. The tails can pack very well around each other, so they tend to be solids, whereas oils are the triglycerides that are liquids at room temperature. They usually have unsaturations among them. So if you notice, again, saturated triglycerols can pack well around each other. Okay, I'll just ask you to imagine many other units of this fat molecule and stack them together. So you should be able to observe that you can imagine them to be very compact, okay, around each other. Whereas for the oils, the glycerides that have unsaturations, you can see that if you try to pack them around each other, there's a natural distance among them. So they tend to be liquids at room temperature. So let's now go to phospholipids. Phospholipids, as previously mentioned, they just have a phosphoric acid group connected to one of the glycerols. So they're also known as phosphoacylglycerols or phospholipids. So glycerol plus two carboxylic acid components plus phosphoric acid forms phosphatidic acid. Okay, so recall again, the carboxylic acid plus alcohol produces our normal ester. Phosphoric acid and alcohol, glycerol, forms a phosphoester. For this particular molecule, it's called a phosphatidic acid. Okay, so glycerol, carboxylic acid 1, carboxylic acid 2, and phosphoric acid. So this is now phosphatidic acid. But it doesn't end here usually. The phosphatidic acid undergoes another esterification with another alcohol to form phosphatidyl ester. Okay, so here's an example of a phosphatidyl ester. Once again, we have the glycerol, carboxylic acid 1, carboxylic acid 2, and phosphoric acid. And the phosphoric acid, if you look at the structure, it also has another hydroxyl group capable of another esterification reaction. So here, the R, which is unspecified in this example, is just another alcohol. So we now form the phosphatidyl esters. Okay. So some examples of phospholipids, you have phosphatidylcholine. So you have glycerol, the two carboxylic acid components, the phosphoric acid at carbon 3, and then the alcohol that is now connected to the phosphate group. Okay, so this makes this phosphatidylcholine. Okay, um, space filling model is shown over here. Another one is phosphatidyl ethanolamine. Okay, so the alcohol that is connected to our phosphatidic acid is ethanolamine. An interesting phospholipid is diphosphatidyl glycerol, which is also known as which is a cardiolipin. Okay, so you may guess where this can be found. So it's very interesting because you have, again, once again, our normal phosphatidic group, two of them, and then they're joined together by another glycerol group. Okay. Now, a simple example is phosphatidylserine. Okay, so we know serine from our amino acids. So serine has an alcohol group, so that can be the alcohol that esterifies to the phosphoric acid of our phosphatidic acid. So again, this is just a survey or sample of some phospholipids that can be found. Okay, so further examples. Phosphatidylglycerol itself. Again, recall glycerol is an alcohol, so that can be the alcohol that's connected to our phosphoric acid group. In the previous slide, we met cardiolipin. And then inositol. Okay, so preview, this is a carbohydrate. But okay, so we'll worry about this later, except that again, notice that there are hydroxyl groups, so any of the alcohol groups can form an ester with our phosphatidic acid. Okay, so I just found this summary table of the different phosphoacylglycerols now. So again, a general structure is you have your alcohol, you have the alcohol, which is the glycerol, two fatty acids, phosphoric acid, and then X, which is again your alcohol, which can be anything. They can be ethanolamine, they can be choline, they can be serine, they can be glycerol, or any other alcohol components. So we're now being introduced into the variety of phosphoacylglycerols that are possible. And why is there a variety? Again, there's a variety of functions, biological functions that they will have.